Okay, good morning. So we have a new session in these sessions of talks in Introduction to Bioinformatics, and today we have with us Rosa Fernandez from the Metazoa Phylogenomics Lab at the Institute of Evolutionary Biology in Barcelona. She's actually the group leader of this group. Rosa did a master's in zoology and a PhD in evolutionary and conservation biology at the Complutense de Madrid. After spending a few years in Harvard, he came to Barcelona as a postdoc, first at Tony Gabaldon's lab, and later he set her own lab at ESCI-UPF, at the Institute of Evolutionary Biology. Her group is interested in using phylogenomic approaches to understand how animals are related to each other, how their genomes evolved to generate diversity, and how they adapted to new environments. Today's talk is entitled Leveraging Phylogenomics to Understand Evolution Across the Animal Tree of Life. Welcome.
Thank you, Arna. Thank you so much for the invitation. It's a pleasure to be here today to tell you a little bit about something that it's not biomedical, but rather application of bioinformatics to basic research, which I think it's a lot of fun. So today's talk will be about this thing called phylogenomics. And I will start by giving you a very short introduction of what this is. And later on, I will tell you a couple of examples of how we can leverage this branch of science to understand animal evolution in the context of the animal tree of life. So let's start by talking about phylogenomics. What is this? So simply put, phylogenomics is the intersection of phylogenetics and genomics. I'm sure that all of you are aware at this point what genomics is and what a genome is, but some of you may not be familiar with the concept of phylogenetics. So let's zoom in a little bit. Let's dedicate 30 seconds to this because I think it's definitely very interesting and needed to understand phylogenomic approaches. So phylogenetics is the science of reconstructing the evolutionary history of all lives on Earth. And the way we represent this is with this type of graphs called phylogenetic trees. They are fully bifurcating, so we have in the terminals some species of interest, could also be genes, could also be traits. And they are separate, they are clustered. Sorry, I'm getting familiar with this. Okay, got it. They are closer to other species or genes or traits that are more similar to each other. And the more far away they are, the more different they are. And we have a few important things in this type of graphs. One is the nodes, like these ones, which are the branch points that represent the ancestors. And then these branches, the longer they are, the more change has occurred in this species. So basically, this phylogenetic tree is a hypothesis of how species or traits or genes are related to each other through evolution. So in this context, A is more closely related to B than to E, for example. And as you can intuitively think, this is very helpful to understand, for example, how are we related to fish or how a butterfly is related to a beetle, right? To understand the animal tree of life, for example. So phylogenetics started a long time ago. There has been many scientists working on that because as you know, there are many drawings from the 18th century, 19th century representing phylogenetics trees, right? So it is a notion of evolutionary relatedness that started early on in the scientific community. And at the beginning, what the scientists were using were morphological traits. For instance, if we want to compare different species, let's say a human, a cat, a whale, and a bat, one can think on which bones within the arms are homologous, similar to each other, and we can start clustering this in a phylogenetic tree to understand how these species relate to each other. So later on, in the 60s, scientists realized that genes have a lot of information because they have nucleotides, right, that encode for proteins that have functions. So they realized that indeed this information could be used as well to build phylogenetic trees. And this, what I'm showing you here, it's only the sequencing of a single gene. This is called Sanger sequencing. It's not that old, believe it or not. I did my PhD with this technique. Now we have genomes, but in the, let's say, 10 years ago, 15 years ago, people were sequencing gene by gene, and the way it works is you put your DNA with a bunch of fluorescent dyes, and each nucleotide gets marked with one of them, and then by the color, you can read the sequence. And then you can have this sequence and compare it with other sequences to build, again, your phylogenetic tree. Later on, pretty much at the beginning of the century, people started to have access to genomes, transcriptomes, massive genomic information. So there was another transition from a single or a bunch of handful genes to hundreds and thousands of genes. And this is what we call phylogenomics. So here, this is just a matrix showing all the species that we have, that would be the terminals in our tree, and each little dot represents one gene. So this one has like almost 1,000 genes. And as you can imagine, this is absolutely fantastic because this is giving us much more information to build this tree, to understand how species or genes are related to each other. So I'm gonna touch a little bit on how we build these trees. This is quite complex, so I'm just gonna discuss very briefly the main rationale behind it for you to understand. I know there are some courses on phylogenetics in the grade if you're interested, but I think nonetheless it's very interesting for everyone to understand how this is done. So in the case of morphology, the way it works is we have our set of species. Just imagine we want to build a species tree of vertebrates, right? And we have lamprey, sea bass, antelope, bald eagle, and alligator. What we can do is to select a series of traits that are informative. In this case, the presence of jaws, the presence of lungs, and having a gizzard or not. And we can use these traits to understand which species have them in common or not. And then we build the most parsimonious tree. This means the minimum number of changes, right? So if we have this tree, we can place the origin of jaws here, the origin of lungs here, and the origin of gizzard here, and will give us this topology. Now we can build other trees, but they will involve more changes. For instance, just imagine that we place the origin of jaws here and the origin of jaws here. That could be another, and this would mean that the sea bass is the sister group to lamprey in the phylogenetic context of this. However, it would imply that jaws have originated twice in evolution, which is less parsimonious. So this is the rationale behind parsimony, one of the methods originally used to build these trees. This was done mostly with morphology. But then, as I told you, in the 60s, people started to realize, okay, we can actually use each nucleotide or amino acid in each alignment as one character, right? Instead of lungs or fur or whatever, I can have amino acid 1, amino acid 2, amino acid 3. I was told that you were actually learning a little bit about alignments, right? So in this context, I think it's easy to visualize that these methodologies for tree building tend to cluster together genes, in this case, that have alignments that are more similar to each other. So we have, like in this example, two main blocks, one here for these sequences, another here for these sequences, and we know that they are more or less similar to each other and the same here. So what happens when we go from a few genes where we can see the alignment and understand how things work, we escalate to hundreds or thousands of genes, so the real phylogenomics. So the process is a little bit more less intuitive because, of course, you cannot be checking gene by gene what's going on when you have 5,000 genes or 10,000 genes. So I'm going to go very quickly through the process, the analytical process that we follow when we want to build these species trees or these gene trees. So we start with the genome assemblies, in this case a human, a fish, and a fly. The first thing we do is to try to identify sequences that could be contaminants. Just imagine that you are touching a sample and your DNA ends up there, so it's easy to map against a human and delete it, or fungus, or other potential contaminants. The next step is something called orthology prediction. So what is this? This comes from the fact that genes duplicate in the genomes, right? This is something that it's well known. We have some genes that at certain point evolve through speciation events, but genes duplicate all the time. And the problem is that if we include all the sequences, including duplicates, we are going to have redundant information that it's going to bias our phylogenetic inference. So what do? Our goal is to infer orthologous genes. What are orthologous genes? Orthologous genes are the ones that evolve through speciation events. So in this tree, we have a frog that contains one gene. This is gene 1. Could be, I don't know, an insulin receptor L1, if you want, just as an example, to put a name. The frog has a single copy, but then what we see is that there has been a duplication in the ancestor of the dog and the human. So we have 4 genes. One copy of the dog is sister group to one of the human, and the other one, the blue one, is sister group to the blue one of the human. So this cluster of blue genes are paralogous to these ones because their most common ancestor is a duplication event, but each of them, they are orthologous to the frog because if we take these two and go back to the most recent common ancestor with the frog, we saw that it's an speciation event. So orthology inference is about going through each of our trees, trying to detect these events, and just getting rid of one of the branches. And this way, we will end up with 3 genes, all of them representing a speciation, which is what we want. I know this is complex, but I just wanted you to have a notion of what it means, because it's the base for phylogenomics. So once we have our set of orthologous genes that we know they have evolved through speciation, we move on with alignment, which is basically trying to put each amino acid or nucleotide on top of each other to later on apply these phylogenetic inference methods. Sometimes we have to identify outliers. It's common that we still have contamination and some duplicates that end up having long branches, and it's desirable to get rid of them. Then once we have the alignments, we may want to get rid of regions that are highly variable. Just imagine that you have different genomes with an insertion or a deletion, and then when you try to align them, they don't match well. Usually what we do is to get rid of that area because it's going to add a lot of noise while we are building the trees. And the last part is either to concatenate them all together to have what we call a supermatrix that will end up having hundreds of thousands of amino acids, and this is massive information to build our trees, or there are other methodologies that analyze each tree individually. And we infer our trees. So I briefly told you before about parsimony, right? That let's say nowadays a little bit outdated, but was very helpful to understand how to build morphological trees. Just to mention that there are two main methodologies to infer these phylogenetic trees. This is not trivial. These are probabilistic methods. And again, if you're more interested, I think there's a class on phylogenomics. I will be also very happy to talk to you if you're interested one day and you want to drop off in the lab to explore this. But basically, these methodologies are two. One is called maximum likelihood, and the other Bayesian inference. And these are a set of algorithms that try to answer a different question. This is just for you to have a notion that there are these methodologies. So don't worry too much, but I just wanted to mention them. So in Bayesian inference, the question is, what is the probability that the model that we are applying is correct given the data that we are observing? Something that I did not mention, but is definitely very important, is that we are going to have a model when we try to understand how similar sequences are and how they have changed. There are a series of models of evolution, most of them are experimental, that give a certain weight to each change in amino acids. Because we know that from a chemical point of view, changing one amino acid by another, it's not equally probable. There are some changes in amino acids that are more easy from a chemical point of view than others, and we have a series of matrices to compare the different alignments and give each change that we observe a different weight. And this is what the algorithms take to build the trees. So given these models of evolution, Bayesian inference answers this one. Given that, what is the probability that my model is correct given the data that I have? And maximum likelihood tries to answer a different question, which is the probability of seeing my observed data given that my model is true? So just for you to have a basic idea about the main methodologies that are out there to build these trees. So this is a very brief introduction to how we do phylogenomics. But something that I wanted to touch upon as well is the origin of the concept, because not everyone knows where phylogenomics come from. Phylogenomics, the term phylogenomics was actually coined very recently, in 1998, by this scientist, Jonathan Eisen. He was using this concept to talk about prediction of gene function and gene family evolution. It was not to build a species tree, it was to understand gene family evolution. And what he realized is, well, we just cannot take sequence similarity and assume that they come from the same ancestor, that if two sequences are more similar to each other, they have the same ancestor, because we know that there are some molecular mechanisms such as convergence, etc., that could be biasing this. And what we need is to build a tree. We need to build a tree, and we need to see if two genes are related to each other. And then we can strongly believe that they are indeed related to each other, rather than just checking how similar or dissimilar their sequences are. So basically what he was proposing is the workflow that I was telling you before. Choose your genes, identify the homologs or orthologs, as I showed you before. Infer the gene tree. Then if you, what he was suggesting, if you know one of the functions in one of the genes that you have in your gene set, by having other genes clustering together, you can assume that these genes have the similar function, right? So this is kind of the notion of using phylogenomics for comparative genomics. But as I told you, our terminals could be species, and one of our main goals may be to infer a species tree, the animal tree of life, the arthropod tree of life, whatever. So it was not until 2002, so 20 years ago, that scientists started to use phylogenomics to infer species trees. And this is the first paper that did it from the team of Hervé Philippe. They were interested in understanding the phylogeny of several amoeba, and they used 123 genes, which was a lot for the time. And basically, they represent another qualitative jump from using just a few genes to using a real phylogenomic approach to understand the animal tree of life. Not animal, amoeba. So as you can see, there are two main big acceptions of phylogenomics. The first one is related to inferring a species tree, understanding a tree of life, the position of one or several taxa in the context of others, and another one that is using phylogenomics to do comparative genomics, to understand gene family evolution, for example. And one important thing is that they are interconnected because, of course, if we want to do something comparative, we need a fully resolved species tree to polarize the results. Otherwise, we don't know when speciation, duplication, or gene loss may have happened. So these are the two main assumptions on phylogenomics. So one of the take-home message that I would like you to take from here is that phylogenomics can be very important to understand the evolution of life forms. And I always like to show this example that you can bring with you and tell your colleagues if they ask, why is phylogenomics important? So one example is this sentence that we all know, which came first, the egg or the chicken, right? So if we take it into a phylogenetic context and we compare turtles, lizards, snakes, crocodiles, and birds, chickens, and we apply phylogenetics or phylogenomics, the answer is very clear. The egg is early, it's first because it appeared here in evolution, and chickens appear later on, right? So it's a clear example of why we need phylogenetic thinking and phylogenomics to understand how traits or species evolved. And with this, I know this was a little bit dense, but I thought it was definitely important to understand how we use these methodologies to answer biological questions. So the second part of the talk is going to be about two examples of how we use these methodologies to answer biological questions of interest. And for that, I'm going to tell you two stories. The first one is really related to the first acception of phylogenomics, that was building a species tree, understanding the phylogenetic position of some species to each other. And I chose spiders because we are, I think it's an appropriate theme for October, being so close to Halloween. And this is a paper that we published a while ago. And the question here was understanding the evolution of the spiders that build this type of webs that you all have seen, right? So spiders are a hyper-diverse animal lineage. They have like around 50,000 species. And a large part of them build these highly intricate webs that they are called orb webs. So one of the most important things is that the behavior to build this type of webs is highly complex. So it's really articulated. It requires a lot of energy, a lot of time. So for people in arachnology, it was somehow clear that due to the complexity of all this, probably the spiders that build this type of webs should be monophyletic. It should have evolved only once in evolution, right? Because it's so complex to articulate a full behavior to do all this. But the thing is that there are two main groups of spiders that can actually build this type of webs. And the difference is in this organ called the cribellum. So there are two main groups. One that have this organ. This organ is like a comb. The silk leaves from the spinnerets, this part of the abdomen, and then it's combed, you know, like when you comb your hair. Some spiders comb the silk and then start building the orbs. And some other spiders don't have it. They just have the silk and that's it. So, and these are some pictures of both types of spiders. I know they look very similar, but they are not. So there's another important difference in the type of silk, because the ones that don't have this organ, they are called ecribellates. They have indeed a different type of silk. It is very sticky because it has some drops of glue. On the others, and it's, for example, one of the most typical ones that you can see pretty much everywhere, right? However, the ones that have this kind of comb, to comb the silk as it leaves, it is sticky because it has a lot of surface. It doesn't have this glue, but it has a lot of surface, and then it's sticky because of that. So there was already some evidence of, okay, perhaps morphologically they are indeed very dissimilar. So again, our main question was, okay, can we use phylogenomics to understand if this type of spiders, called orb weaving spiders, are monophyletic or not? And this will help us understand whether the evolution of this highly intricate behavior to build the webs evolved once or twice. And for that, what we did is to go to the field. I was telling Arnaud, I didn't realize, but I should have put here some pictures of field work, because in my lab, we actually go to the field, we collect the creatures, we do the wet lab, and then we do the bioinformatics as well. So, you know, evolutionary biology can be a lot of fun, and phylogenomics can be a lot of fun as well. And once we had the spiders, in this case, we sequenced transcriptomes, because it's cheaper and, than doing a genome, much easier to compute, and at the same time, we have thousands of genes that we can analyze. And we ended up with a set of 2,100 genes. We ran a bunch of analyses. Something that I would like to touch upon very briefly as well is that analyzing a lot of genes comes at the price of having to do many different analyses, because by concatenating information, we are also exacerbating different biases that can be affecting our data. So when you do a phylogenomic analysis, you don't do a study, you don't do one analysis, you do like 20 or 30 or 40. And this is what this figure represents. So basically, we were taking the 2,000 genes, we were concatenating them together, we were also using them, analyzing them individually through different approaches. We were accounting for some factors that we know that are affecting phylogenomic inference, such as, for example, missing data. We are not recovering the same genes in all the species, so when we concatenate all the genes in all the species, we end up with a matrix that is like a Gruyere cheese that has holes, right? And this can bias our results, or the evolutionary rate. There are some genes that evolve faster, others evolve more slow. So we need to also check how this is affecting, and so on and so forth. And what we got is this. Let me guide you through this tree here. Basically, we have a bunch of spiders. The ones we are interested in are these ones that are the ecribellate, and this one in green that you cannot see well, that are the ecribellate ones. And in each node, we have a bunch of little squares. These represent all the analysis that we did, the 21 analysis. And if you see color, it means that there was a very strong support in every single analysis for that. So when we see something like this, it's like, okay, all the analyses were congruent with that node being strongly supported, so we can believe that. So what our results told us is that indeed, remember that phylogenetics trees are bifurcating. This is not, you cannot see it well because of the colors, but this is not the sister group to this. There was some doubts because different analyses were telling us different positions, whether they are the sister group of these other spiders or these other spiders, but definitely they are not the sister group to these spiders. So we could believe with strong support that orb weaving spiders are not monophyletic. And the arachnology community was really surprised about that because, again, this means that the evolution of that super intrincated behavior occurred at least twice, which is very interesting. So now let's move on and talk about beetles, because something that Arnau did not mention is that we have a second line of research in the lab, which is understanding the evolution of cave animals. I'm going to tell you a second story. This we published recently this year, and this was spearheaded by Pau, a former PhD student in my lab that finished this year, and who loves caves. And what happens in cave fauna? Cave fauna are white, they have elongated appendixes, they tend to lose the eyes because they live in a place that is dark, has constant humidity, and they need to rely more on touching than visual cues, right? And I'm showing you here different animal lineages, completely very divergent, and they all tend to have these phenotypes, this white color, this loss of pigmentation, this loss of the eyes, this elongation of the appendices, etc., compared to their surface sister species. And our study system, in this case, was beetles. And why beetles? Because beetles are indeed excellent models to understand subterranean evolution. So if we represent the beetle tree of life with its main branches, beetles, by the way, is one of the most diverse lineages of animals on Earth. It has more than 400,000 species. So when you see a beetle, it's 1 out of 400,000 species alive. They have these two main lineages called Adephaga and Polyphaga. And it is great because it has many different instances of lineages that ended up colonizing caves. So it means that we can compare surface and cave species in independent colonizations, which is great to understand how the genetic repertoire changed to adapt to caves. So thanks to a collaborator, we got some cave species from 3 different lineages in Australia. They are from these tribes, Videsini and Hydroporini, and they are aquatic. This is the type of environment where they live. So you basically put like a bucket and bring water up and hope that you have beetles to collect them. And this is the surface environments where we got the surface ones. And this lineage colonized caves between 12 and 4 million years ago. On the other side, we went to caves in the Pyrenees and in Slovenia and Greece to collect terrestrial cave beetles, in this case from this tribe called Leptodirini, and with older colonization of caves, like around 33 million years ago, compared to the 12 million years ago of the other species. And as you can see, they have, I mean, you see some color, but they are much whiter compared to the surface ones. And they have very elongated antennae, very elongated legs, and most of them have very reduced eyes, and some of them have actually lost completely the eyes. So we wanted to expand our study and include many other beetles as well, to understand with more precision which gene families changed convergently in all these independent colonization of caves. So we ended up having representatives of 6 colonizations. This is the aquatic lineage. In black, we have the subterranean species, and in white, the surface ones. So as you can see, we have 1, 2, 3, and 1, 2, 3, minimum 6, which is an excellent system to understand which gene families we could observe that are common or not in these transitions. So again, our aim was to investigate the genomic basis of this adaptation to subterranean environments in both terrestrial and aquatic beetles. And what we did is to go to the field, go to the lab, end up with many transcriptomes, again in this case, and a good representation. This is kind of the backbone of the beetle tree of life, and we were just marking the representatives that span the full tree of life of Coleoptera. So we have a good representation of what's going on within the full order. And we ended up with almost 15,000 gene families to analyze. And what we did is to apply phylogenetic comparative methods to understand which of these gene families changed in a similar manner in each of the 6 independent colonizations, either by expanding their copy number or reducing their copy number. So one of the first things that caught our attention is that most of the changes that we observed were not in the ancestors of the surface and cave ones. Let's take, for example, these 2 species that are sister to each other. This is a surface one, this is a cave one. Not many changes occur here. The proportion of the changes here are represented by the size of these circular sectors. So the bigger they are, the more change in the gene repertoire. And in orange and green, it's expansions rather than loss. Because something that I did not mention is that what the scientific community thought is that since these animals tend to lose things, they lose pigmentation, they lose the eyes, etc., perhaps this means a loss of genes involved in these functions. And the first thing that got our attention is, no, these animals are not losing genes. They are indeed gaining genes. And moreover, these gains are not occurring in the ancestor that separates each surface and cave one, but occur before. In every single case, before, look at this. Like here we have another example of surface one and caves one. There's a little bit more here, but most of the changes occur before the diversification of each surface and each species that colonize cave afterwards. And the same occurred with gene duplication. When we were understanding how many genes duplicated, we saw that most of the duplications occur before these 2 sister species, the surface and the cave one, diverged. And this is very interesting. Here it says major changes in the genetic repertoires. This is very interesting because this is telling us that something occurred in the genomes of the ancestors that could mean a genomic acceptation later on. So probably, we don't have full genomes, but probably these genomes underwent a whole genome duplication or an exceptional expansion of certain gene families in the ancestors, and later on, independently, the different cave beetle species could use these new extra pieces in their genetic repertoire to adapt to these environments. So we were defining this concept of cave species lose genes. No, they don't. They are very similar to the surface ones. What happened is that in the ancestors, there was a boom, an expansion of the gene repertoire, and they could use differentially some new pieces to adapt to these environments. So now we zoom in to compare each surface with each cave one. And what we saw is that the gene families that were expanding or contracting were exactly the same. This is what we refer to in this context as parallel evolution. However, in the aquatic lineage, however, in the terrestrial one, what we observed is that each tribe was doing something completely different. So this means that for each of the 3 independent colonizations, the surface and the cave were expanding or contracting completely different gene families, but the functions were very similar. So it tells us a little bit also about genomic redundancy. Having gene families that do similar things can be also good to adapt to different environments. And between the terrestrial and the aquatic ones, we also detected convergent evolution. So different gene families underwent expansions and contractions. So just to give you a glimpse of the putative functions of these gene families, what we observed is that most of the families that were expanded, so new genes doing potentially new things, were involved, contracted, that were lost, were involved in the immune system, which makes sense because you are exposed to different viruses, bacteria, fungi, etc., in caves compared to surface, right? Lipid metabolism, what we believe is that these animals tend to use different sources of energy in caves compared to surface because food is less homogeneous, right? You need to hunt, animals, you need to hunt for your sources in a space that doesn't have a lot of availability. Photoreception, which is kind of straightforward because you don't have light, so that was kind of good to see that indeed genes involved in response to light, such as photoreceptors, were lost. Chitin binding, so the chitin is what makes one of the pieces that makes the cuticle, so this is telling us that the cuticle changed. And circadian rhythms dependent on light as well. There are some genes in the circadian rhythm that are independent from light, and these were present. However, the ones that depended on lights were lost as well. And another surprise was about the genes that were expanded or in a similar manner in the different lineages. One of the categories had to do with transposases, so this is transposable elements. This is giving us a hint that perhaps transposable elements had an important role also in adaptation, but we cannot go further than that. Signal transduction, and more importantly, many gene families that we don't know the function because there's no single gene that has been experimentally validated in other species, and we just simply don't know what they do. And we also saw chitin binding. So this is telling us that, for example, the cuticle the gene repertoire that was involved in the cuticle was completely reshaped through gene loss on one side, but also gene gain on the other side. So just a few take-home messages about these stories are a little bit more complex than the previous one because the questions were more complex as well. So the genomic changes that facilitated adaptation to life in caves across beetles showed both parallel and convergent evolution, so parallel in the aquatic ones, in the aquatic beetles, but convergent in the terrestrial ones. Gene loss was remarkable in beetles that colonize subterranean habitats, mostly in genes involved in photoreception, circadian rhythms, and so on. But on the contrary, gene gain and duplication was key in reshaping the genomes, particularly through expansion events that occurred in the ancestors. And aquatic beetles underwent remarkable changes in their gene repertoire, indicating more recent genomic remodeling, and surface-dwelling and subterranean terrestrial beetles experience gene repertoire changes prior to the diversification of the tribe, indicating in both cases perhaps genomic exaptation through this expansion of the gene repertoire. And this is it. Again, the goal was to give you an overall glimpse of what phylogenomics is and how we can use it to answer specific biological questions, such as understanding the spider tree of life and the evolution of this intricate or weaving behavior on one side, and on the other, how the gene repertoire has changed in beetles to adapt to caves. And I hope you had some fun. And this is my group, our sources of funding. We are at IBE, across the street for PRBB. So if you are interested in any of these topics and would like to come and say hi, you are most welcome to do that. And I will be happy to answer any questions. Thank you.
Okay, thank you very much. Very nice talk, very interesting stories.
Thank you.
So now let's go for the questions. Before that, I should warn you that the take-home message that Rosa showed are the messages that she would like you to take home, but these are not necessarily the take-home messages that you have to write. Okay. So first questions from those on the priority list.
You have explained how phylogenetics help to answer biological evolutionary questions, but it is always possible to know what conditions caused these bifurcations between species, for instance, with two species with very small difference of DNA?
So phylogenetics help us understand the hypothesis of how things are related to each other. However, if we want to understand the cause, we need to use other type of methodologies, definitely. For example, in the case of gene repertoire evolution, it is now really valuable to have chromosome-level genomes to understand where genes that are duplicated are placed exactly, to see how are they regulated, if they are close to similar regulatory elements or not. That can help us understand a little bit the molecular mechanisms behind that, definitely. And what you are saying about the NE, this is kind of the interface between phylogenetics and population genetics, which is something that if you have a question that refers to that, you may need to apply methodologies from both fields to have a more complete picture of your question of interest.
Well, what are your team goals from now up to the future?
What are my?
Team goals, your investigation achievements for the future.
As I now mentioned, our main topic of research is animal terrestrialization. So what we are doing is to generate many high-quality genomes from different animal genomes, phyla that we know that left the sea and conquered land. So let me go a little bit back. There are 36 animal phyla. Some of them are well known, such as vertebrates, right, that are subpart of a phylum called Craniata. Arthropods, we have seen some examples here. You have annelids, you have mollusks, but then there are some others that are not that well known. For example, nemerteans, ribbon worms, tardigrades, velvet worms, and so on and so forth. I'm just throwing some names here for you to open your mind about the extent of the question. So out of these 36 animal phyla, 10 of them originated in the sea. All animal life originated in the sea, but 10 of them managed to colonize land. And just imagine suddenly going to Mars and having to learn how to breathe, how to move in a different gravity, how to be exposed to different radiation levels, and so on and so forth. So this is more or less what happened to them. Not such a sudden episode, probably it was little by little, but they needed to smell on Earth, on air versus smelling on water. They needed to eat different things. They needed to repair their DNA from UV lights. They needed to learn to move with more gravity. They needed to deal with much more oxygen levels, and so on and so forth. So what we are trying to do is to generate genomic resources for each of these animal phyla and compare the marine ones with the terrestrial ones to understand how the genomes reshaped to be able to adapt to these new conditions. So to put it simple, we are trying to understand the genomic basis of the origin of terrestrial animal biodiversity. And a secondary line, we work on cave animals, but this is a little bit more secondary.
You talked about all these studies with beetles and spiders, so that they have a large lineage. So in this case, how do you choose which species do you want to work? How is this process?
Yeah, this is actually an excellent question because all the inferences that you do in phylogenomics are valid in the context of the species that you include. So you have to usually do some preliminary research about the distribution, how animals are distributed in lineages, to take representatives of all of them. Because if you take, let's say that you want to do a study of vertebrates, but you only take fish, then you are not going to have a good representation of the most deep nodes, right? Ideally, you want a good representation of mammals, amphibians, reptiles, birds, and different fish lineages, right? So you definitely need to search to understand the evolutionary context of what you want to answer, and find good neighbors around your species of interest or your lineage of interest that give you enough depth in the tree to go back to the nodes and try to see what changes occurred. So yeah, this is what we call taxon sampling, and it's definitely very, very important.
We know that databases such as the National Library of Medicine are used for studies, but are there any specific study groups around the world that focus on each species one by one and then put them in common? Because, well, I guess that the amount of data that is needed for phylogenetic trees is huge.
Can you repeat? I'm not sure I understood everything. Can you repeat the question?
I'm asking if, is there like specific study groups around the world that focus on each species one by one in order to create these phylogenetic trees? Because I guess the amount of information you need to create these trees are huge, and there are not like all the information you need.
So making a link with your colleague's question before, it is extremely important to choose taxon sampling of the species closely that are good neighbors to the species that you want to place. Because orthology inference is going to be strongly determined by how divergent the species are. If you want to compare an amoeba with a human, you will end up with not many orthologs. Because the node you're trying to resolve, just imagine you put all the species on Earth and you build a super giant tree of life. The node you're trying to illuminate is deep, 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 deep down. So it would not make a lot of sense, right? So if you want to compare, I think the most important thing here is to have clear in your mind what is the question. In our case, it was, for example, this example. We want to compare this group of spiders with the other group of spiders. So I need spiders from both groups, and then I need spiders from the other groups that I know they are closely related. But it doesn't make any sense to put a mollusk there, for example, because I know it's very divergent. It's not going to add a lot of information, and we are going to lose many orthologs in the way. So I think it doesn't make a lot of sense to do a giant phylogenetic tree with all the species on Earth. I think it makes sense to tackle specific questions and zoom in specific lineages of interest. And this is also related to something that I did not mention here that is more related to the algorithms. But the algorithms of phylogenetic reconstruction work better with not so many tips, because of course the alternatives that you have to evaluate exponentially increase. So if you have millions of species, you have to evaluate with these algorithms like probably trillions of species trees, and this is extremely complicated. So
Thank you.
Can we use phylogenomics to predict how a certain species will evolve?
I don't think so, but you can use it to identify how unique they are. In the sense that just imagine that we have 10 spiders and 8 of them are very similar to each other. and then 2 of them are very, very divergent, very separate. Their genomes are completely different. This can tell us that this species probably evolved in a very separate and different manner than the other ones. So it can tell you that they are a bit more special. And this can guide some type of, perhaps, conservation actions and things like that. Just imagine that these 2 species happen to be on a very tiny island. Then it's like, okay, this tiny island has 2 species of spiders that are extremely different compared to the other ones that we have. So they are precious, because the other 8 ones, or 8,000, are more similar to each other, but these 2 ones are very, very different. And we want to preserve forms of life that are different, right? So I don't know if this answers your question more or less.
Oh, yeah.
But yeah, I think you cannot predict evolution forward. It's easy to look backwards, but I think looking forward, it's definitely complicated. But you may try to guess how important your species are depending on your question of interest.
Okay, any other question? Yeah.
How did you study the better specimens that you collected from all around the world? Did you need special permission to take them, or you studied them in the country where you collected them?
Yeah, that's this is another seminar completely, has enough content for a different seminar, because of course you need a permit for everything you collect, and you are not allowed to just go and collect the species. You need local permits, you need national permits, you need export permits, you need import permits, so life gets a little bit complicated, but of course it is the way to do things. We have to stick to following the international legislation, of course. And just in case, just to, to mention something very interesting, a few years ago, most countries in the world signed a treaty called Nagoya, the Nagoya Protocol, that's making things a little bit more complicated, because this is to avoid that people from like other countries go to your country, even if they have a permit, collect the material, and then they take a benefit out of the genomic resources that have been produced. Just imagine, for example, people going to the Amazon. That is a hotspot of biodiversity. No, I don't know, I'm a rich pharma company. I go there, collect a few frogs, and I extract some venom, and then I patent it, and then I gain millions of euros, and the people from Brazil get nothing, right? So, I mean, it's complex, it's legal, it's ethical, it touches on many different aspects. And now you need to get a Nagoya permit, meaning that if you get a profit out of what you're doing, you need to give back somehow to the country. But each country has its own legislation for it, so it's extremely complicated. Yes?
So I wanted to ask if you have discovered any new species when you go to any field and find animals. And
indeed. So I did my PhD with earthworms, and we described 7 species and 3 genera of earthworms. And we actually, very recently, like a few months ago, we described a species of a phylum that is very neglected. They are called Nematomorpha, horsehair worms. In the Montseny. It was the 6th species described in Spain, and we sequenced the genome chromosome level. So the paper is a mixture of the description of the species and the description of the genome. And it's the first genome for the full phylum. So it's kind of interesting and exciting, because at the end, we can apply bioinformatics to do very relevant discoveries about biodiversity. And this is very cool.
Okay, so I think it's a good moment to stop with this excitement. Okay, thank you very much again.
Thank you, everyone.